So welcome everybody. I want to present to you some work we have been doing over the past years. It is a follow-up. We have made a paper about 4 years ago on the effects of the herbicide glyphosate on plant, animal and human health. And now we are looking at more at the indirect effects of the herbicide glyphosate on health through its effects on microbial communities. So the indirect environmental effects we have discussed before. We are a group of people led by Professor Dr. Arina van Bruggen, who is a professor emeritus at the University of Florida, and a few other colleagues here from China and Wageningen at the Netherlands. And we have been working on this topic, as I say, for a while. I want to first look at what is glyphosate, look at the mode of action, the formulation, and then how it fades in the environment. And then I want to look in the main part on the effects of the microbiomes in health, talk a little bit about what is the microbiome, how we analyze it, and then go on to the health effects, and finally finish with the conclusion. Glyphosate is the active ingredient of the herbicide Roundup and others. There's about 70 glyphosate-based herbicides. This changes over time. It is N-phosphonomethylglycine, which is a derivative of the amino acid glycine. It was developed as a disinfectant by, back then I think it was called Ciba-Geigy, but it was not strong enough, and so they gave the product to Monsanto, and they accidentally discovered that it has herbicidal activity. The mode of action of glyphosate is by blocking the shikimate pathway. It is an important pathway for plants to produce tryptophan, phenylalanine, and tyrosine, and basically all aromatic metabolites and proteins that contain aromatic amino acids, which are these three. Part of it is, when we look at plants, it's lignin, tannin, flavonoids, but also many hormones are affected by this. It is crucial, this pathway, for plants, bacteria, fungi, protozoa, and algae, which means the product does not only affect plants, but also other organisms. Glyphosate not only blocks this pathway, but it also has direct antibiotic properties, and this is directly taken out of the US patent text, which says that as antimicrobials, these compounds may be expected to induce stasis rather than cell lysis or death, allowing the infection to be cleared by the host's immune system. Such an outcome is desirable, they're right, as it will ameliorate the absolute selective pressure to select for the growth of resistant mutants, which would inevitably be the case if the compounds used caused cell death. This I found a little weird, because if we are prescribed an antibiotic, we are always asked to take it all the way to the end in order not to select for resistance. And any antimicrobials that are not strong enough will, of course, also select for resistance. So I'm not so sure if this claim here is really as benign as is being said here. A formulation means that all chemicals that we apply in agriculture have to be formulated so they can be really applied. First of all, we have to ensure correct solubility. So glyphosate salts are used rather than glyphosate itself, so it can be dissolved in water. But also, what is very important, most plants have a waxy surface, and it is very difficult for liquids to stay on the plant surface and not to just run off. So normally tensides are being used to improve the contact with plants and to reduce the surface tension of the water. And the one surfactant that has been used in the EU and worldwide is polyoxyethylene amine, toluamine in German, and it's abbreviated usually with these 4 letters, POEA. It is prohibited in the EU since 2016, but not in the rest of the world. In the EU they prohibited it because it is very toxic. And this is an example. One of these, this formulant, as far as I know, is toluamine, but we have seen others with formulant like this. What you see here is tomato plants that have been sprayed either with water, with glyphosate alone, or with 3 types of Roundup, meaning glyphosate-based herbicides, or with the formulant alone, which means without glyphosate in it. And what you see is that after 120 hours, which is 5 days after application, the tomatoes are not yet dead. This is normal because glyphosate takes 5 to 10 days to kill the plants. However, if glyphosate is formulated, here it is much faster, as you can see, and even the formulant alone seems to be more toxic than glyphosate. And this is one of the problems we encounter with the glyphosate-based herbicides. Nevertheless, glyphosate itself will kill the plants if you wait long enough. So the formulants alone are also herbicidal, and if the package indicates rapid action within 1 or 2 days, then you can be sure it's not glyphosate that has been killing your plants, but it's something else, because glyphosate takes 5 to 10 days. When they started using glyphosate in the 1980s, it took about 0.7 kilo per hectare to kill all the plants. This is 0.07 grams per square meter. It's not very much. Currently, the prescription is already to use 1.1 kilo per hectare, which means 0.11 gram per square meter, so about 50% more than before, because overall the effectivity of the chemical has changed. If the plants are destroyed, all the animal habitat is gone. What you see here in the US, they started with less than 0.5 kilo per hectare in the 1980s. By now, the recommendation is 1.8. So they even apply more glyphosate than we do here in Europe. The metabolite is aminomethylphosphoric acid. AMPA is toxic. What happens if you apply glyphosate a lot and all the time? Glyphosate resistance arises. Here you can see in the picture the field has been sprayed, but there is green plants, and these plants are resistant to glyphosate. If you look at the Wheat Science website, you find glyphosate resistant weeds worldwide, and I took this in 2018, and you can see from 1995 there was no resistance to glyphosate. Until now we are over 40 species are resistant. The last one here on the list is Lactuca saligna. That was in 2018. I went back to the website and checked now 2021. You see here Lactuca saligna, that was in 2018, and there's several more have been added to the list of being glyphosate resistant. This is not very surprising, as the glyphosate resistance per se is naturally occurring in certain plants. And the genetically modified plants that are glyphosate resistant, they have basically a naturally occurring gene for glyphosate resistance has been built into these plants. Glyphosate use is very high worldwide. I could not really find the actual data for the world, but we have data from Benbrook in 2016 who showed that since 1974, about 8.6 million tons of glyphosate were used until 2014, and 72% of this in the last 10 years. In 2014, use was 826,000 tons worldwide, and that was in the US. They used about 1 kilo active ingredient per hectare cropland, which means total cropland. Many parts of the cropland do not receive glyphosate, so where they apply it, they apply more. Worldwide, on average, 0.5 kilo per hectare are applied. In Germany, I could find the data. About 1/3 of the cropland are treated every year. And in 2020, we used about, on this cropland, 0.66 kilo active ingredient per hectare. So if it's 1/3, we used quite a bit more. What happens in the environment? That depends. If spraying is done before planting, you would see some weeds that will, of course, be sprayed. You may get some washout to the groundwater. It will evaporate, and there is wind distribution via the soil and dust. There is some root uptake of glyphosate through the roots of plants. So if glyphosate is in the ground, plants will take it up. This is why you have to wait after spraying for a while. Then it would be transported in the plants and may even get into the pollen if it's GMO plants. A second way of using glyphosate is what we call seccation. That means spraying before harvest. In a lot of places in this world, this is done routinely to kill off the cereals before harvest in order to make them mature at the desired moment. And that means in a moment when the cereals are not totally ripe, they are sprayed with glyphosate, and glyphosate is transported in the plants down and upwards, and also through the roots, released into the ground. And on the other hand, of course, we have residues in the ripe grain if it has been treated with glyphosate. Through the atmosphere, of course, they can also be washed out by rain, and it can also get into the surface water. In soil and water, glyphosate is degraded to AMPA. We have here the whole glyphosate. Here you get CO2, and this is cut off, and it is AMPA. This is done by bacteria. In genetically modified plants, there's an extra gene so that the decomposition of glyphosate starts in the plant, and AMPA is being released from the plants into the ground. In former times, glyphosate was released into the ground with strong effects on the microbiome. AMPA itself is very slow in decomposition and also accumulates in water, and it has the same toxicity profile as glyphosate, and has been shown that it is cancerogenic. You can see here the time it takes for glyphosate to be reduced. Glyphosate is found in soils worldwide wherever people look, in relatively low amounts. It's all that is green. When we get then to the orange, we have 0.1 to 1 milligram per kilo of soil in the top 30 centimeters in the year 2015. So, but you see, it is worldwide. The most heavy use is in North America, Brazil, a little bit down to Argentina, and then of course Europe and East Asia. There has been a German study that was released last year, and it showed that glyphosate is really distributed in the air. I translated it here, that all samples of non-biological origin, that means coming out of the air, through passive samplers that just collected what comes in with the air, contained glyphosate. If you look at a map, you can see here the red is glyphosate, the blue is AMPA. And in these passive samplers, they found glyphosate all over the place, and they also found AMPA. And they found it not only where glyphosate is being applied, which is heaviest in northern Germany, as you can see here, but they also went to nature protection areas, and wherever they looked, they found glyphosate, which actually documents the aerial dissemination. And we have data from Sweden, from areas where never ever glyphosate has been applied nearby, and you will find it. Also in the soil, many times people have claimed that glyphosate binds to the soil particles and is not washed out into the ground, but there are data from a long-term monitoring in central Germany, and they went down to 4.8 meters in the ground, and here are the data for glyphosate, and you can see that they found glyphosate also in very deep down in the ground, which shows that it is transported down into the ground. The problem when glyphosate gets into deep zones is there is no more microbial degradation, and that means that the glyphosate then can get into the groundwater without being further degraded. This was published in 2020, and you see here the maximal concentrations that were found in microgram per liter, and you see glyphosate was the highest concentration that was found in the surface waters in Germany, followed by some others, but glyphosate was by far the most found. In 62.7% of the cases where they looked, they found glyphosate. In about 40% they found AMPA, and 9% they found other chemicals. And there is a minimum, maximum, and mean, and you can see that the mean micrograms per liter for glyphosate also was dramatically higher than for all the other chemicals. That's enough for the environmental fate of glyphosate. Now I want to come to the main topic, the effects of glyphosate on microbiomes in health. First, I want to talk about the microbiome itself, what it is. Then, how do we measure this? And this was one part that we have been looking at in more detail in this paper. And then the new disease dysbiosis that is emerging in animals and plants. Then look at glyphosate and antibiotic resistances, and on the overall effect of glyphosate on the microbiome in the soil, rhizosphere, and plants. I need to go a little bit back, and what have we learned in the recent past? We have been learning that by now we are living in the so-called Anthropocene. That means the era that is mostly influenced by humans. And there was a very nice paper by Sariola and Gilbert in 2020 when they wrote that on the one hand, microbes are defined as the foremost enemy of humans. This is what many of us think when we think microorganisms, we think diseases. In our failed attempts to eliminate microbes from our lives, the Anthropocene has created the conditions for recombinant viruses and antibiotic-resistant bacteria. On the other hand, we have recognized that microbes are part of our very being. And this is so important. Yeah? We are not only talking about pathogens, but we are mostly talking about microorganisms that we need. Our health depends on the symbiotic bacteria that helps build and maintain our healthy bodies. The body's immune system has evolved to recognize the difference between benign and potentially pathogenic bacteria. So what they write further is that by now we do not think about single organisms anymore. We rather think about what we call now the hologenome. The hologenome theory recasts the individual animal or plant and other multicellular organisms as a consortium, the holobiont, that the host plus all its symbiotic microbes. While we inherit about 22,000 genes from our parents, we inherit about 8 million different genes from our parents' bacteria. This means that a lot of the genetic information that is playing a role in our bodies does not come from human genes, it comes from microbial genes. So now the question is, who is the microbiome? We know about 9,000 bacterial species. The estimation is that there are about 10 to the power 6 to 10 to the power 9 species that exist, and most of them we don't know. So instead of 9,000, 10 billion. Most of the bacteria can only be found through DNA analysis because we cannot culture them in the laboratory. The few we see in the laboratory are very, very few of the whole bacteria. The DNA usually enables the idea of empires, kingdoms, classes, or families. In some rare cases, we can also identify species, but those are very few. In most cases, it is a very rough method. So if you look at this, this is just part of the bacterial phylogeny, and you see all these names: Acidobacteria, Actinobacteria, Aquificae, and so on. And you see there's many different groups. So I give an example which is maybe easier for everyone to look at the leguminous plants. The Fabaceae is a legume family, and we have about 20,000 species that all belong to this family. And within these species, we have more than 700 genera. Basically, the species are divided into about 700 genera. So if DNA analysis can tell us the genera, we are already quite good. But oftentimes in bacteria, it's only the family that we are being told. So when you look at papers that describe bacterial communities, you very often find alpha proteobacteria, beta proteobacteria, gamma proteobacteria, and that is a very wide group. So the question is, what are we actually looking at? And so how are people actually quantifying microorganisms if they cannot count species? For example, one thing that has been done for a long time is to measure the microbial activity. So we measure metabolic processes, how much CO2 is created, how much O2 is used, how much whatever is being metabolized. Then microbial biomass can be looked at. There we can then calculate, okay, if there's so much biomass, we can, on average, say there are so many single microbes around. Or we can search for known species, so at least we get an idea. We can classify according to the higher taxonomic orders, as I just described. And just look at the proteobacteria, which is a higher order. Within that, we have many alpha proteobacteria, beta proteobacteria, gamma proteobacteria, etc. And just looking into this one group of alpha proteobacteria, you have there good and bad guys. Brucellosis is not a good idea to catch this pathogen. Rhizobia, very important for nitrogen fixation. Agrobacterium can be a pathogen. Rickettsia usually are pathogens, and so on. In the beta proteobacteria, you have Ralstonia, a severe pathogen, Nitrosomonas, which are important in nitrogen cycle in the ground. And then gamma proteobacteria, you have Escherichia, but also Shigella and Salmonella, Yersinia, the pest pathogen, and so on. Pseudomonads that are very often important in biocontrol. So just saying I have alpha proteobacteria does not tell me anything about who do I have. Another important thing is when we look at how many do we have or how many species. Just an example, I designed 2 microbiomes. Both of them have the same number of individuals and even the same species, the red and the green. So the same individuals means probably the metabolic activity and biomass are almost the same. And the same species can be found, the green and the red. So if we look at this level, we will not see a difference in these 2 microbiomes, which is obvious, however. The equilibrium is shifted. And this is something we start paying more attention to when we think about microbiomes. There are alternatives, for example, to quantify genes that are connected to specific functions, nitrogen cycling, manganese reduction, antibiotic resistances. They give us at least an idea how frequencies shift. Do we have more or less of something? And now I come to the dysbiosis, the new cause of disease. As I pointed out before, we have a lot of microorganisms inside our bodies. The estimates are 90 to 90% of the cells found in a human being are microorganisms. Dysbiosis are deficient or aberrant microbiomes, and they are more and more recognized as the underlying causes of diseases, including mental problems. And the use of antibiotics changes the microflora. Everybody who has taken antibiotics hopefully has been told by the doctor also afterwards to eat a lot of yogurt and lactobacillus in order to rebuild the microbiome in the intestine. And glyphosate is patented as an antibiotic, and this is where the connection. So the question is, could it be that we get dysbiosis through glyphosate? This symbol here indicates changed microbiome. So if you have a plant that is treated with glyphosate, the microbiome around the plant roots may be changed. The residues of the plant may get into the animals, and this may change the microbiome inside the stomach of the animals. And the residues may also accumulate in animal products and get into humans, or directly into humans. And also here we can get, we can ask, do we have problems with our microbiome because we consume glyphosate? In addition, of course, we have the runoff of the glyphosate into the surface water and leaching into the ground. Some experiments have been done, for example, on rats, looking at what happens if the rats consume glyphosate within the allowed residue levels that are normal in water. So baby rats whose mothers consumed in the drinking water the allowed residue levels of glyphosate had a changed microbiome. For example, the pathogen Prevotella was increased and lactobacillus was reduced. And lactobacillus usually is a beneficial. Female young rats produced more testosterone and sexual maturity was delayed. And the mothers and babies had neurological and behavioral changes related to the changed microbiome in the maternal intestinal tract. And if they were fed lactobacillus and bifidobacterium, the memory improved. If we look at the antibiotic glyphosate and what can it do to the microbiome, one very important example is, for example, the bee colony collapse disorder. That has been going through the press for the last 10 years. The problem that we have a lot of bee decline, a lot of bees dying, colonies suddenly disappearing, especially in North America, but also in Europe. And so people started doing research on this and looking into what is causing bee colony collapse disorder. And one question that people started asking was, could it have to do with glyphosate? So some people in Texas did some very interesting research. There are several papers continuing all the way into 2020, and I'm presenting just a few of these data to make the point. So what one needs to know is that neonate bees, the ones that are emerging from the pupa, are sterile. And they get fed with regurgitated. That means that what worker bees go out, feed, and then they basically regurgitate what they have eaten to the young bees to feed them. And with that, they inoculate the young bees with the intestinal bacteria that they have in their food and that they have in their guts. And so the question was, what are the effects of glyphosate on the intestinal flora of bees? The experiment was done like this. Here you see a neonate bee emerging from the pupa. They took them out directly at this moment from the beehive and fed them for 5 days, either with sterile glucose or with sterile glucose, including 5 or 10 milligram per liter glyphosate. And I will talk about this concentration in a moment. They said this is realistic in the United States in GMO plants you may find this much glyphosate in the nectar. After 5 days, they put the neonate bees back into the beehive so they could be fed by their colleagues with what the colleagues have in their guts. And then they looked at the intestinal flora. So you have the control group, you have the bees that get 5 milligram per liter glyphosate, and the bees that got 10 milligrams per liter glyphosate when they, at the moment before they return to the beehive. No difference in the intestinal flora. Then 3 days in the beehive, 3 days being fed by their partners, the control group had significantly more bacteria than the group that got 5 milligrams of glyphosate per liter, but not the group that got 10 milligrams of glyphosate per liter. So there was some effect, but not totally clear. So there was some reduction of bacterial growth, but it was variable due to antibiotic effects of glyphosate, was the conclusion. Then they did a second experiment. They again took the neonates for 5 days, and they fed them either gut homogenate of the they fed them with gut homogenate of the workers, but they added to the gut homogenate glyphosate at 10% of the concentration of in experiment 1. So they went down 1 order of magnitude. And so they had, with and without glyphosate, they fed them gut homogenate. And then they looked at what does the intestinal flora look like. And they could show that in the control, the intestinal flora was different from the glyphosate treated one. You see some shift in the flora. One of the typical pictures, there's some shift, but we do not know who is who. So, and then they went to the third experiment. They looked at what happens to the bees when they are challenged with a pathogen that is called Serratia marcescens. So again, they got gut homogenate or sterile water, sterile glucose, or they got gut homogenate with or without glyphosate, or sterile glucose with and without glyphosate. So a total of 4 treatments. What you see here, gut homogenate plus glyphosate, gut homogenate without glyphosate, microbial free, which means sterile, and microbial free plus glyphosate. And you can see over time back in the beehive, you have a little bit of mortality, but 80% still are alive after 8 days. Then they added, so all treatments survived equally well in the absence of Serratia. Then they added Serratia here. You can see here the SER. And the gut homogenate treated bees that got gut homogenate, no glyphosate, but they were challenged with Serratia, about 50% died. So you have a significant death rate due to Serratia in the normally fed bees with gut homogenate. If the bees got gut homogenate with glyphosate or sterile sucrose with Serratia, or sterile sucrose with glyphosate and Serratia, basically all of them died. So you can see that the gut homogenate is protecting them rather well if it is not contaminated with glyphosate. So glyphosate obviously destroys the protective function of the guts. Now let's go back to these concentrations, 5 and 10 milligrams per liter of glyphosate, that they were starting out and then they went down even an order of magnitude from this in the second and third experiment. But what does it mean? So in Germany we apply 1.1 kilo per hectare. So let's take 1 kilo. We apply it in 200 liters of water. That means we apply 5 milligrams per milliliter, which means 5 grams per liter. So 5 and 10 milligrams per liter is really 2 orders of magnitudes less. So 500 to 1,000 times less than the concentration applied to plants. It sounds reasonable if you think if it's taken up in a genetically modified plant and then transported into the nectar or so, you expect that the concentration is less than what you applied. This is relevant, thus, for genetically modified plants. But what about when we spray plants in order to kill them, as we do in Germany, and they are not genetically modified? So we apply 5 grams per liter. In the first step, the concentration rises because the spray dries up. And if in that moment the plants that are not yet dead are being visited by bees, which is normal, we have weeds, they are flowering, they are sprayed with glyphosate in order to disappear, then the bees come and feed also on the liquids that are on these plants. They get an extremely high dose of glyphosate compared to the experiments we were just looking at. So bees that visit non-GMO or GMO plants shortly after they received an application and are not yet dead, also the non-GMO don't die within no time, yeah? They will be exposed to much higher concentrations than tested by Motta et al. In the paper that I presented. And so we find this on demonstrations in Germany. It says, too much glyphosate. We have to throw away 4,000 kilo of honey. We have enough of the poisons of agricultural industry. I took this photo a couple of years back. And this is not a fairy tale. This is reality. The farmers had to dispose of this honey. And so it is absolutely correct that farmers who think about this ask that glyphosate should not be applied to flowering plants, because the bees do not distinguish if it's a weed where they shouldn't fly to or if it's a normal wild plant. Let's go to the question glyphosate and dysbiosis on humans. There's a paper that was published 2 years ago by a group in Canada. They are just asking, do we maybe have a problem with glyphosate that is different from what we expect? So they did a nice review of the literature surrounding glyphosate, dysbiosis, and weed sensitivity. I do not know what is the situation in Bosnia, but around here, and especially in North America, a lot of people claim they cannot eat wheat well. They claim that it gives them problems in their bowels, in their intestine. So they checked what does glyphosate do to the microbiome. If you have glyphosate residues, the Spirocheta, Fusobacteria, Prevotella, Actinomycetes, all go up. These are the arrows that say they are increased with glyphosate. Butyricoccus is decreased. Then if you use glyphosate-based herbicides, the list is larger because you have the adjuvants, the formulants, and you can see a long list of organisms that are either increased or decreased. And then there is here in the middle you have Ruminococcaceae and Lactobacillus that are decreased as well in glyphosate alone or with glyphosate-based herbicides. So this is the background. So they looked at the data. They took the data from 2009-10 and looked at the people who are avoiding eating gluten. 100% of the people who avoided gluten were divided into groups that either had celiac disease, that was 5% of them avoided gluten because they have celiac disease, which is a genetic condition and that cannot be changed. Another 51%, they actually had celiac disease, but they didn't know. They were later tested for it, and then it was clear, aha, you're avoiding gluten because you are a celiac. So you have 56% avoided gluten simply because they had a genetic condition that made it impossible for them to eat gluten. But 44% avoided gluten, although they did not have the genetic disposition. So 44% that avoided gluten did not have celiac disease in 2009-10. Then they looked 4 years later, and there very few people who avoided gluten did not know that they were celiac. So a total of 28% were celiac, and more than half of them knew it already. But at that time, 72% of the people who avoided gluten had no celiac disease. That means the proportion of people that claim they cannot eat wheat is increasing dramatically, despite the fact that we certainly do not have an increase of the number of people with the genetic disposition of celiac disease. So what's happening, they were asking. So they were looking at how much do people eat, yeah? And they looked at the glyphosate residue allowance in the USA, which is basically the same as in Canada. So people, the Canadians, eat 67 kilo of wheat, a little bit of corn, a little bit of soybean, some canola per year. So wheat is really a large part. They looked at how much glyphosate is applied to wheat. Not as much as in corn and soybean because they are genetically modified, but still. And then they look at the maximum residue level in North America. And they took the data from 2015 from Health Canada. Unfortunately, they were wrong. When you look at the paper by Ben Brook, that was also from 2016, he already showed how the residue levels have changed. You have here residue levels that are allowed in the USA in feed given to the animals, and this goes up to 500, and ppm is milligram per liter per kilo, and or for food, for human consumption, it goes only to 50. So you can see for animals, they get a lot more of this than we do. And if you look at the different crops that are looked at here, you can see with wheat in 2015, the residue levels went up from 5 to 30 milligrams. And that is dramatic. So since 2015, 30 ppm glyphosate residues are allowed in wheat for food purposes. Not, as the Canadians thought, 5. And in animal feed, 100 to 400 ppm are allowed. So this is really dramatically higher. The reason that they do it, that they have increased the values, is because they got into this habit of spraying wheat dead before harvest, and they could not get the residues at 5 ppm anymore. So they simply raised the residue levels. If we look at the glyphosate residues in plant products, in urine, you find concentrations depending on the crop of up to 120 or 85 milligrams per kilo. And the maximum residue limits depend on country. And you can see some allow 0.5, some allow 40 milligrams per kilo. From 0.1 to 30, from 1 to 40, depending on crop. In feed, the residue limits that are allowed are always higher, and the residue levels measured are also dramatically high. This means 1 gram per kilo, 1099. And accordingly, human urine samples, 40 to 95% of the samples that are tested, depending on country, have glyphosate residues. And the concentrations up to 3.2 micrograms per liter. So this is an order of magnitude less, of course, than what we find here. But still, 0.001 is 1 microgram. And so up to 3.1 micrograms means 0.003. It comes up to what we find in the crops already. And the limits, of course, the maximum concentrations documented up to 233 micrograms per liter. That means 0.2 milligram. So it's dramatic. So the maximum allowable residues in products were raised with the increase in detected concentrations. Residues are generally higher in North and South America than in Europe. Clear, because a lot more GMOs are grown there. In the USA, residues are still sometimes higher than the US norm. And the high percentages of the human urine samples are contaminated with glyphosate. Let's look at some disease data from the United States. That was done in 2014 by Swanson and co-authors. They looked at a total of, I think, 30 different diseases and disorders in the US databases, and then they made correlation graphs. So in this case, for example, it's intestinal infections due to microorganisms. In the late 80s, the death per 100,000 was down here, and it was relatively the same over time, and then it slowly increased. And you have to know that in the late 80s, glyphosate started being applied, yeah, but not very much. In 1996, GMO glyphosate resistant corn and soybean were started to be grown in large scale, and this is where you see the glyphosate rates go up. And in parallel, the rates of intestinal infections, the death rate with intestinal infections is also going up. The same with diabetes. There was an overall uptrend all the time, which means people are not eating well anymore, eating more and more sugar, more and more carbohydrates. We know that this is a main problem with diabetes. But still, despite this trend that was already ongoing, since the introduction of GMO, and you can see here the blue line is the GMO corn and soybean crops grown, and this is the glyphosate being applied. At the moment these come in, diabetes goes up. The same with autism. Again, children with autism are dramatically going up since the late 1990s. So putting these all three together, and then they did it with about 30 diseases, they everywhere found extremely strong correlations with 0.96, 0.98. But of course, all these diseases that I listed here can be caused by dysbiosis. But a correlation does not mean glyphosate is the reason. Anybody in this lecture audience that eats carrots will die, and that does not mean that carrots are deadly. So correlations have to be looked at with big care. So the authors of this study, and me too, I say we need to research to find out, is there anything connected? Is this connection real? Are these correlations true, or can we refute them? They did some statistical tests, of course, that showed if you have so many correlations all going in the same direction, it is highly unlikely that glyphosate is not part of the picture. But it's a correlation. Dysbioses are overall increasing. So we have this study here, genetically engineered crops, glyphosate, and the deterioration of health in the United States of America, clear correlation with glyphosate use. Another study, Clostridium difficile infections, an overview. He shows that there's a connection with the use of antibiotics and the rise of Clostridium. Richard all show that there's a connection with pseudohormone active chemicals, with the intersex variation from toxicological dysregulation in fetal productive tissue. Then Aguilera et al. Show a connection with change in the microbiome and development, looking at the influence of microbiota disrupting chemicals in relation to specific endocrine pathogenesis. Endocrine pathogenesis means hormone-related diseases. So we really need to look at this. This is not just a fantasy of some people. And so let's look back. It is an antibiotic. So now let's look at how does glyphosate and microorganism interact. We have this EPSPS pathway, the shikimate pathway that I presented before, that is responsible for the production of the aromatic amino acids. And there is two different, by now we actually know we have three different pathways to produce this amino acid. There is one pathway that is glyphosate sensitive, and there's one pathway that has an increased glyphosate tolerance. If you add enough glyphosate, you will also stop that. And if we look at that, we find that we have high glyphosate tolerance in some really bad pathogens: Clostridia, Fusobacteria, Salmonella, Clostridium tetani, Orthobacter brucellosis, many Fusariia, and Helicobacter, which is the causal agent of the bacterial ulcers in the stomach. In addition, many plant endophytes are highly glyphosate sensitive. That means with glyphosate residues, we favor pathogens and we may disfavor important endophytes. And we know, for example, in the human gut, a large percentage of the gut microflora is glyphosate sensitive, and Lino et al. There give a nice overview on that. And that means it is clear this will influence animal and plant health. So if we look at autism, for example, autism correlates with glyphosate-tolerant Clostridium species. That means they have shown that children with autism have a lot more infection with glyphosate-tolerant Clostridium species. And so what does it mean? If we look in agriculture in general, we have veterinarian medicines that make input of antibiotics. We have antibiotic active herbicides. It's not only glyphosate, it's also dicamba and other herbicides. And the herbicides are applied on a large scale worldwide. So we may have an open circulation of antimicrobial resistance between antibiotic resistance in individual microbiota, antibiotic resistance in local groups, animals, and environments, and antibiotic resistance in nations, continents, global natural environments. And this is only one part of the picture. I'll show you the rest. We know we have cross-resistances to antibiotics. For example, glyphosate and antibiotics have been shown to react with cross-resistances for penicillin, for ciprofloxacin, and kanamycin. Those are three different types of antibiotics. They all interact with glyphosate. And one study showed then that after glyphosate exposure, Salmonella were more resistant against ciprofloxacin and kanamycin. This is dramatic because it means that we may be increasing the multi-resistance by using glyphosate and other herbicides. A very interesting study I ran across was a study that was entitled "Manure from Animals Enriches Taxa that Commonly Carry Resistance to Beta-lactam Antibiotics." They compared what happens to the soil when they fertilized with NPK fertilizers or with manure from animals that were not treated with antibiotics. So they used experimental animal herds where they knew exactly that these animals had not received antibiotics, so they knew it was not coming from veterinary medicine. But of course, the animals in the US, they eat cereals that were desiccated, and they eat GMO soybean, and they eat GMO maize in their ration. That means they have high residues of glyphosate in their ration. So if it is not veterinary medicine, as they showed here, where do these resistances come from in the manure? They finished the paper by just saying, we don't know. If you think about what the animals eat and what kind of straw they use, if they get straw at all, then you can imagine that in the manure you have quite a bit of and quite a bit of glyphosate residues, and the beta-lactam antibiotics are part of this game. So Korenbach and colleagues in New Zealand, they wrote a paper a couple of years back, and they wrote here as one of the significant findings that neither reducing the use of antibiotics nor discovery of new ones may be sufficient strategies to avoid the post-antibiotic era. And if we think about the current COVID-19 epidemic, we should recognize that we have in parallel a pandemic of antibiotic-resistant pathogens running for more than a decade already. And nobody's talking about this very much. But good hospitals will test their patients when they come in if they carry multi-resistant bacteria, because this is one of the major issues within hospitals that will cause people to die. So if you look at the whole picture, we have virtuous circles in individual health, one local health, and global health. They all are connected. Yeah? We need to understand the concept of one health via microorganisms. We have globalization processes, we have overpopulations, we have frequency of migrations, travel, and interactions. We have a decrease in animal and plant diversity. We have much more contact with the wild because we are infringing on the wild, and COVID-19 coming from wild animals that we basically should not be eating. Huh? We have too many opportunities for microbiota to get together where they don't belong together. Then we have heterogeneity in sanitation facilities. We have pollution and the damage to natural ecosystems. All this functions together and makes then that we get this circulation of the antimicrobial resistance throughout. And so if we want to have a better future with less pandemics, we have to think in this way and think about the microbiome also. So in a paper we published a couple of years back with the title One Health Cycling of Diverse Microbial Communities as a Connecting Force for Soil, Plant, Animal, Human, and Ecosystem Health. This is also depicted. And you can see how we all are connected via our excreta. Yeah? It goes into the soil and the water, and there it goes through the plants, it goes to the silage, it goes to the animals, or directly to the people. And if we then apply stress, everything changes. So a quick look at glyphosate effects on microorganisms in soil, the rhizosphere, and plants. We have changes in microbial communities on and in plants through glyphosate. For example, growth-promoting pseudomonads are reduced. These pseudomonads are typical manganese reducers. They make manganese available to the plants. And that means there's reduced nutrient availability, and that leads to increased fertilizer needs. I have seen in Brazil people using a lot of iron fertilizers, foliar iron fertilizer, in areas where the plant nutrition people in Brazil said, we don't have a problem with this. But if they use GMOs, they know they need it. So what is the result? The plant nutrition is compromised. And this is an accidental find by the University of Hohenheim in Germany. They had fields where they regularly used glyphosate here for 11 years. And they had next to them 2 fields that for some reason they started the glyphosate use 9 years later. They had only 2 years of glyphosate application. And then they realized that plant growth was massively inhibited in the years that had gotten glyphosate for long term. And this is how the people from the University of Hohenheim started looking in more detail, and they also went to Brazil to check what does this long-term application of glyphosate do really to plant nutrition. And they could show that a lot of plant nutrients are blocked, and the composition of the plants that are grown like that is also different. You have different levels of zinc, of iron, and other things inside the plants, and that makes a difference. Bacterial microbiomes on the root surface have been checked lately. So in this paper, this is the gene abundance of soybean Z31 that was treated with glyphosate. And this is a gene abundance of soybean Z31 that was not treated with glyphosate. And you can see the untreated with glyphosate had a different microbiome than the treated one. Certain overlap, of course, all that is brown, but the blue are only for the untreated, the yellow ones only for the treated ones. So many metabolic genes and genes of individual organisms were more prevalent after application of diluted glyphosate on leaves or genetically resistant soybeans. And some functional genes, plant growth promoting genes and N fixation, in organisms were less prevalent after glyphosate treatment. So they went in, what I said before, they checked for certain functions. Are they more or less prevalent in these communities? And so they found plant growth promotion and nitrogen fixation were reduced by glyphosate application. And this means more fertilizers have to be given. I have had a phone call from a German salesperson who told me 10 years ago already that they have been told you can sell Roundup for very cheap because you will sell so much more fertilizer and pesticides afterwards that it will be absolutely worth your while. So they knew exactly what they are doing. Glyphosate effects on Fusarium severity on GMO maize here. So here's the glyphosate application between 0 and 300 micromoles per liter. I cannot translate that in the glyphosate dosage. Sorry, I have not done my homework for that. But you can see two pathogens, Fusarium verticillioides and Fusarium oxysporum. And you see with increasing glyphosate use, there's an increase of disease severity. The interesting thing was, when they did it in Petri plates, both of these Fusarium pathogens were also slowed down in their growth by glyphosate. So glyphosate, these pathogens also suffer from glyphosate, but less. The difference was that they grew faster on root surface because they had reduced growth of and reduced competition by pseudomonads. And the pseudomonads are the biocontrol ones, and they are more sensitive than Fusaria to glyphosate. So it's this interaction. If you just check in the Petri plate, you see Fusaria are also glyphosate susceptible, but not as strongly as pseudomonads. Just to remind you what is actually happening. Plants and microorganisms belong together. Plants take up CO2, they release O2, they make assimilates out of this, and they release a lot of the C into the ground. The estimates are between 30 and 60% of what is assimilated is released into the ground in C-containing substances that feed the bacteria and fungi and other microorganisms. In exchange, the bacteria and the microorganisms, the fungi, they give nitrogen, phosphorus, and sometimes also water back to the plants. If we have glyphosate-treated plants, we have an increased secretion of C and amino acids, and that attracts microorganisms. We have reduced competition for pathogens due to microbiome shifts. We have reduced production of defense-related metabolites in the GMOs because we have these aromatic amino acids that are not being produced, and they are part of the defense-related metabolites. And potentially we have changes in the associated endophytes, especially in the GMOs. So in summary, what we can say is glyphosate is found everywhere in the environment, in the air, in the soil, in the water. It accumulates in the environment. Glyphosate is an antibiotic that causes microbiome shifts in the soil, in plants, and in animals, and thus also humans. And changes in the composition of the microbiome can lead to dysbiosis. The antibiotic glyphosate causes cross-resistance against several important classes of antibiotics: penicillin, ciprofloxacin, kanamycin as examples. And what are the consequences for agriculture? We have increased disease susceptibility, especially to soil-borne diseases, and more for GMO plants than for non-GMO plants. We have reduced nutrient availability if glyphosate is applied long-term. We have increased need for fungicides and fertilizer as a consequence. We have disturbed intestinal microflora and diseases in animals, and thus also in humans most likely. We have increased antibiotic resistance in the environment. And thus, in conclusion, there's not much to say. But we have in Europe the precautionary principle. That means you have to check ahead of time for the risks. The connections between glyphosate, diseases, including dysbiosis, need to be investigated. And the large-scale application of an antibiotic like glyphosate and some other herbicides is simply insane and needs to be banned. With this, I just want to present to you my co-author, Professor Dr. Arina van Bruggen, who has been instrumental in putting together all this work, and would open for questions now.